answers, check it against your answers, raise questions. What is true about amylase? Both saliva, salivary glands, and pancreas produce amylase. It's important for the digestion of carbohydrate. What's true about pancreatic secretions? All of them. Questions about either of these clickers? Gastric acid. What have I already told you about gastric acid? I haven't taught you this, but it turns out it's both to hydrolyze protein and to activate an enzyme in the stomach that is important for the digestion of protein. Questions about that? What's the function of bile? Emulsify and solubilize fat. What volume of saliva? 2 liters. What stimulates the secretion of saliva? Parasympathetic nervous system. During the cephalic phase of digestion. I can increase your production of saliva right now simply by talking about food. Right? If I talk about, oh, the lasagna I'm going to have for dinner tonight, it's so good. I can smell it. I'm producing my own saliva right now. Just thinking about food, but now making you think about food, even if you don't like lasagna. Something good, you are now salivating. That's the cephalic phase of digestion. Your head is in it. Okay, so those are the clickers. Any questions about those? Okay, what I'm going to do now is turn to swallowing, and then peristalsis in the esophagus, and then we'll get to the stomach. So I'm actually going to show you a couple of YouTube videos. First one is about swallowing, or deglutition. Well, these are both about swallowing. The first one's about 2 minutes, and the second one's about a minute.
Swallowing, or deglutition, is the process by which food passes from the mouth to the stomach. This movement is controlled by the nervous system and involves both voluntary and involuntary muscle contractions. Before food is swallowed, it is mechanically broken down in the mouth by chewing or mastication. The salivary glands secrete the enzymes lingual lipase and salivary amylase. Salivary amylase begins the chemical breakdown of starch, carbohydrates, in the mouth. The water content of saliva helps bind particles together to form a bolus that can be swallowed. Swallowing occurs in 3 stages. The first is a voluntary phase called the buccal or oral transit phase. Here the tongue moves upwards and backwards against the hard and soft palates to push the bolus into the oropharynx. Next is the pharyngeal stage, in which involuntary movements push the bolus through the pharynx and into the esophagus. Movement of the bolus stimulates receptors in the oropharynx, which send impulses to the deglutition center in the medulla oblongata and the lower pons of the brainstem. The brain signals the soft palate and uvula to close off the nasopharynx, and the epiglottis to seal off the larynx. This prevents the bolus from entering the respiratory tract. The upper esophageal sphincter relaxes to allow the bolus to move into the esophagus and contracts to reduce backflow into the pharynx. In the final esophageal stage, the bolus is pushed onward by peristalsis. a progression of involuntary wave-like contractions of the circular and longitudinal smooth muscles of the esophagus. As the bolus approaches the end of the esophagus, the lower esophageal sphincter relaxes and the bolus moves into the stomach. So, in summary, food moves from the mouth through the oropharynx and esophagus and then into the stomach.
Okay, that's that one. Any questions about that? This is not what I wanted to show you. Lost the other one. Okay, so what is this? This is an X-ray of someone swallowing a radio-opaque food material. So we're going to actually see data of something being chewed and swallowed. So this is a lateral view. Obviously, mouth, esophagus is in here somewhere. Chewing. Looks like it might be gum or something, but then chewing, making a bolus, moving into the back of his mouth. It happens pretty fast. Peristaltic movement through the oropharynx, nasopharynx, into the esophagus, not into his respiratory tract because of the closure of the opening of the upper esophageal sphincter and closing of the, well, I can't remember it. It's the word for the thing that closes at the top of the respiratory system. The epiglottis. Thank you. Okay, so what that cartoon showed is a pretty good representation of what swallowing is. And the narrator said something about voluntary and involuntary movement. You are born knowing how to swallow liquids, right? You're born knowing how to swallow milk. That's what you start, your diet is liquid when you were born. You learned how to swallow food. Any of you have watched a baby learn how to work baby food in their mouth and get it down their throat, know that it is a learned process. They're really bad at it until they learn. How do you control these muscles in the tongue? How do you control the muscles in the jaw to chew and move the food where it needs to go so that it goes out the back of the mouth instead of out the front of the mouth? And that's a learned process. That's a voluntary process. It becomes, it becomes, you don't think about it anymore, but you had to learn it. So innate and learned muscle function associated with swallowing. Peristalsis, contraction of smooth muscle of the esophagus, moves the bolus, B-O-L-U-S, bolus of food, that's what we make in our mouth, a bolus of food that we swallow, and peristalsis moves it from the mouth through the upper esophageal sphincter, down the esophagus. The lower esophageal sphincter opens and allows that bolus to enter the stomach. The stomach has some anatomical terms that I want you to know. They're on this slide. The esophagus, that's not part of the stomach, the esophagus. The lower esophageal sphincter. The cardia at the junction between the lower esophageal sphincter and the stomach. The fundus at the top of the stomach. the body, the antrum, the pylorus, and the pyloric sphincter at the end of the stomach. Those are anatomical terms. The stomach is going to empty into the duodenum. Duodenum, some people say. Either way is correct. It's going to empty the food, the now digesting food, into the duodenum through the pyloric sphincter. Remember, sphincters are a circular muscle that open, relax, or constrict to close. And those control the movement of food through the digestive tract. We'll see a couple of key sphincters. Rugae is the name for the folds inside the stomach. So the body and the antrum and the pylorus, and to a lesser extent the fundus, all have folds called rugae. And anatomy to learn. So more anatomy, some of which we've just covered. So this was a game that used to work where I could run this on the internet, and people could make, drag this word over to one of these buttons, and it would make a very pleasing ding sound if it was correct, or an sound if it wasn't. But it no longer runs. They've killed the website, so I just have stills. So the body. So, I don't know, maybe this is the body. Closer to the antrum. Maybe this is the body. So but that's a term you should be able to know. This is the fundus. This depends, so if it's talking about the outer surface would be the serosa. So on a quiz I would have to say surface. Just like in the kidney quiz I said surface of the kidney and you told me capsule. On this, if I said surface, you would tell me serosa. If I said area of the body, I would make sure it was pretty clear. It was either in the fundus or in the body. This one is labeling a muscle layer. So this is longitudinal muscle, circular muscle, and oblique muscle layers of the stomach. This is the antrum. It's trying to illustrate some folds, maybe rugae. So I might say folds, and you would tell me rugae. Or I would say area, and you would tell me antrum. There's going to be some structures that we won't see on the cadaver because we've sort of removed them. The lesser omentum and the greater omentum are fatty structures that sort of drape over the digestive tract, and they are connected to the stomach. So the lesser omentum connects to this part of the stomach, and the greater omentum connects to this part of the stomach, and it's like a fatty drape over the intestines. The inner curvature of the stomach has a name. It's called the lesser curvature. Lesser curvature, so you can predict what this would be called. The greater curvature. The lesser curvature of the stomach, greater curvature of the stomach. So if I had a label pointing to this, it could be talking about the serosa, it could be talking about the body, it could be talking about the curvature. I would have, if I wanted one of those answers, I'd have to tell you. I mean, not tell you the answer, but give you a clue. More anatomy. Questions? Okay. There's a word I want to introduce. When we swallow a bolus of food into the stomach, and it starts to get digested by the enzymes like pepsin and the acid, the gastric acid, it is no longer food. And it really is no longer, it's not just a bolus, it's being mixed with all of that gastric juice in the stomach. And it is now called chyme. So I'm not going to be talking about food anymore. I'm going to be talking about the digestion of chyme, because that's what works its way from the stomach into the intestines. So chyme is what food has now become. And you know what chyme looks like. Vomit, right? When you vomit, you're vomiting chyme. No comments from you. The stomach churns the food into chyme. So the circular, longitudinal, oblique muscles form this churning action. The sphincters are opening and closing. Well, the upper esophageal sphincter is now closed because we do not want to have any reflux of chyme into the esophagus. So the upper esophageal sphincter is closed, and the pyloric sphincter opens briefly to allow pulses of chyme to be released into the duodenum. Pulses. So it opens for a moment, a couple of seconds, and then closes again to allow a small amount of chyme to move from the stomach into the small intestine to regulate the flow and not overload the ability of the small intestine to digest the chyme. So pyloric sphincter opens on a sort of a rhythm to allow chyme to slowly move into the small intestine. And there's another video that I forgot to load, but I want you to see. This is gastric motility seen from the inside of the stomach. So this is the inside of a stomach that has been sort of cleaned. It's clean and pink because there's no chyme in it. There is only acid and mucus. So it's empty, but it still has been stimulated to go through gastric motility. So take a look at this. Too distracting. Stupid music. So watch now, a wave of contraction of the pyloric of the antrum, a wave of contraction, grabbing a bolus of chyme and squeezing it out through an open pyloric sphincter. And then it relaxes and does it again. So there's this thing in your stomach, sort of grabbing a bolus of chyme and then squeezing it and then doing it again. So there's this wave of contraction. It looks so alien. It looks so alien, but it's inside your body. Okay, so that's. Okay, so gastric motility, that's what it actually looks like. The cartoon doesn't really do it any favor, but peristaltic action, moving the chyme around the stomach until finally a bolus is squirted out the pyloric sphincter into the duodenum. The stomach is lined with a slightly specialized version of that generic lining that I told you about. So we have the mucosa, muscularis mucosa, lamina propria. You could label this, you could improve the labeling of this slide. So in the mucosa, the epithelial cells with some endocrine and exocrine and mucus cells. So here is a blow up of a fold, which in the stomach has a name. It's called a gastric pit and a gastric gland. The pit is the opening, and the gland is the structure. And the gland has endocrine cells. Well, let's see, I'll stay with their label. Mucus cells secreting mucus. Epithelial cells. Parietal cells that are going to be essential for the secretion of acid. So parietal cells, so epithelial cells are just generic cells for secretion and absorption. Mucus cells make mucus. Parietal cells make acid. Sheath cells make pepsinogen, which is going to get converted into pepsin. And the stomach has some endocrine cells. They're actually called enteroendocrine cells, but we'll just call them endocrine cells. They make a hormone that we're going to talk about in a few moments. Mucus, acid, pepsinogen, a hormone. In the gastric gland that is lining, which is the description of the epithelial layer of the stomach. Lamina propria, muscularis mucosa, submucosa with the blood vessels and some lymph and some pyloric, what's the word? Network of neurons. Plexus, neural plexus. Then the three muscle layers, circular, longitudinal, and oblique. And on the outside, the serosa. So that's the slightly specialized layering structure of the stomach. We've already talked about mucus, so I'm not going to show you those hagfish videos of mucus. It's the same mucus that you line your airway with, more or less. It comes from the mucus cells. So in the airway, what was the name of the cell that produces mucus in the airway? Ciliated cells move the mucus. Goblet cells produce the mucus. In the stomach, they're called mucus cells. It's easier. Mucus cells make mucus. Let's not worry about the spelling. Mucus is a polysaccharide. What is the function of mucus? To coat and protect the stomach. So let's go back one slide. You'll notice that there are stem cells in the stomach. The cells of the stomach are constantly being attacked by their own acid and by the pepsinogen, but by the pepsin. So the stomach cells are constantly under attack by a very harsh environment. And so the epithelial cells of the stomach are turning over and are being replaced by cells that evolve, that come out of a stem cell niche. So the stomach lining is one of those areas of your body that has a very robust set of stem cells. Your skin, stomach, liver, a few parts of your body have a large population of stem cells for regeneration of those cells when they get degraded. Mucus helps protect them, but it's not a complete protection. Let's talk about acid secretion from the parietal cells. Let's get oriented here. Here's the lumen. So what membrane is this? Capable membrane. Here is a capillary in the submucosa. So this is the lamina propria. This is a cartoon of the lamina propria. So this is a capillary in the submucosa. So what membrane is this? Basolateral. Do you suppose we're seeing all the things that are important in a parietal cell? Not even close. Sodium potassium ATPase is expressed on this membrane, as you would expect, but we're just not seeing it. The acid, the hydrochloric acid that is produced and secreted into the lumen of the stomach is secreted by a proton ATPase, a proton pump. We've seen this before in the kidney tubule when we talked about the way that protons are secreted from many segments of the tubule. Same enzyme, pumping protons into the lumen. This slide happens to illustrate in another point, is that it's a proton potassium pump, but we don't have to worry about potassium. It's just not the important function. So like the sodium potassium ATPase that's moving two things at the same time, the proton pump is moving two things at the same time, but the important thing is the proton. Protons are pumped, and chloride follows through chloride channels to make hydrochloric acid, gastric acid. Where does this proton come from? It comes from where you would expect it to come from based on this slide. It comes from carbonic acid. So carbon dioxide, which diffuses there because it's 40 millimeters of mercury everywhere, partial pressure everywhere. Carbon dioxide gets there by diffusion, associates to carbonic acid. What enzyme? Oh, there it is. Carbonic anhydrase accelerates that reaction. Dissociation, there's the proton. I don't know why they put this here. You could just cover this up, cross it out. The proton is pumped into the stomach lumen. The bicarbonate is pumped into the blood in exchange for chloride. So this is the chloride bicarbonate exchanger that we saw in the basolateral membrane of some regions of the kidney. Here it is in the basolateral membrane of the parietal cell of the stomach. So you eat a big meal. What happens to your pH? While you are digesting this big meal. Thanksgiving, let's think about Thanksgiving. I'm making saliva happen, but that's not the goal here. I'm getting you to think about what happens when you eat this big meal. You've got a lot of chyme in your stomach. It's producing a lot of gastric acid to digest all that turkey or whatever other protein you ate. What happens to your pH? The pH of your body goes up because it is becoming alkaline. The pH of your stomach goes down, and the pH of the rest of your body goes up. It's got a name. It's called the alkaline tide that is associated with the gastric phase of digestion. Tide. T-I-D-E. Just like, acid goes in and acid comes out. The water, I won't test you on that. That's how we make gastric acid. It comes from CO2, carbonic anhydrase, making a proton that is pumped into the lumen, and bicarbonate that is pumped into the blood by an exchanger, or by a primary active transporter, or by a secondary active transporter, using some of the language from Selkirk. Chloride follows passively, and that's why we have hydrochloric acid. We don't have to worry about potassium. Questions about gastric acid production?
What happens to people that have acid reflux, and do we treat it by inhibiting the pump?
Acid reflux can be treated by inhibiting the pump, and we're going to talk about that in the context of gastric ulcers in just a minute. Reflux is often a mistake of the lower esophageal sphincter. If the lower esophageal sphincter does not close all the way, then the contents of the stomach can get churned back up into the esophagus. That causes acid reflux and heartburn. Heartburn is just an inflammation of the esophagus. It's got nothing to do with the heart. It's an inflammation of the esophagus as a result of acid reflux. So it can be treated with a pump inhibitor to make the contents of the stomach less acidic. Okay, so now let's turn our attention to the enzymes that the stomach uses to digest protein. So the parietal cells that we've just been talking about make acid. The chief cells that we are introducing now make pepsinogen, a precursor to pepsin. So just like angiotensinogen is a precursor to angiotensin, pepsinogen is a precursor to pepsin. So the cells make the precursor. The precursor is subject to acid hydrolysis in the lumen of the stomach, and that makes pepsin. Pepsin is a protease. It hydrolyzes peptide bonds. So it does two things. It also hydrolyzes pepsinogen to pepsin. So there's a positive feedback loop going on here. Acid gets it started, and then pepsin and acid continue the process of converting pepsinogen to pepsin. And then pepsin hydrolyzes the protein in the meal. So this is endogenous protein, and this is the protein that you ate. But it starts to hydrolyze the protein into polypeptides or oligopeptides. Let's not worry about intrinsic factor. You can cross that out. So this is the story of the secretion of pepsinogen, which is turned into the enzyme for the initiation of the hydrolysis of protein, digestion of protein. So we talked about acid production by parietal cells, and we've talked about secretion of pepsinogen by chief cells. How are these things controlled? The cephalic phase of digestion I told you about is in your head, right? It's your brain telling your telling your salivary glands to get to work. That is the parasympathetic nervous system. We're now talking about the gastric phase of digestion. So we have moved from cephalic to gastric in your stomach. And the gastric phase of digestion is controlled by 4 hormones. Well, really 3 hormones and 1 neurotransmitter. Acetylcholine, the same neurotransmitter that the salivary gland responded to make saliva, is important for the production of acid in the gastric phase in the parietal cells of the stomach. So this is a really bad cartoon of a parietal cell. It's really, really bad. What membrane is this? That's the apical membrane facing the lumen of the stomach, because that's where the acid is going. What membrane is this that is facing blood that would receive endocrine hormonal signals? This is basolateral. So this is a very, very bad cartoon of a parietal cell, because it's not circular, it's cubic, cuboidal. And this is the basolateral membrane, and this is the apical membrane. The point is that hormones like histamine and gastrin increase the exocytosis of vesicles and increase the expression of the proton pump in the apical membrane. So there is this pool, large pool of vesicles containing the proton pump waiting for signals to trigger exocytosis. And the exocytosis puts the pump in the membrane, and the pump gets to work pumping acid. So this is a little similar to that aquaporin story in the collecting tubule that was waiting for vasopressin signals to get exocytosed. The hormones or neurotransmitters are, so ACH is the neurotransmitter that stimulates this. Histamine is a hormone that stimulates this. You may have heard of histamine, its effects in your nasal, it helps you make mucus. Antihistamines help you stop making mucus. Histamine is an important hormone that partially controls this. Gastrin is a hormone that is produced by the endocrine cells of the stomach. So the stomach endocrine cells make gastrin. The name is kind of helpful. It's a hormone from the stomach. Well, you know that gastric acid is from the stomach, so if you know gastric acid, you're 3/4 of the way to knowing gastrin, the hormone from endocrine cells of the stomach that tell the parietal cells of the stomach to exocytose the proton ATPase. So one set of cells of the stomach, let me go back again. One set of cells of the stomach is telling another set of cells of the stomach to get to work, but the communication is through the bloodstream. It's not direct, it's not paracrine, it's endocrine. Here is a slide that is a better depiction of a parietal cell. So these sort of monstrous looking cells, looks like a head of a dragon or something. This is a closer representation to the way they look. This is the apical membrane, and this is the basolateral membrane. At rest, the apical membrane has a certain surface area, it's pretty large, and when it is stimulated, remember stimulation put all of these vesicles into the apical membrane, and so it becomes even more folded. So this large surface area of apical membrane is pumping acid into the lumen of the stomach. So if you see a cartoon with these dragon or lizardy or whatever, this scary looking cell type, that's a parietal cell. And this is just showing that this population of vesicles with these red circles, which are the proton ATPase, waiting to be exocytosed, the stimulation by gastrin, acetylcholine, and histamine triggers that exocytosis, inserting the pump, pumping of proton, following of chloride, hydrochloric acid. Water is also moving here too, because water follows solute just like in the kidney. And in this case, the solute is hydrochloric acid. there's an inhibitory signal called somatostatin, another hormone. Somatostatin inhibits this process. So we've got positive and negative endocrine and neural control. So let's see how that works in a flow chart. Your brain tells the enteric nervous system to make acetylcholine. Acetylcholine is the key. Part of this is the parasympathetic nervous system. Acetylcholine is one of the positive signals that tells the parietal cells to pump acid, to put the pump in the apical membrane and pump acid into the lumen of the stomach. So that's the easy one. Acetylcholine, so label your slide. This is acetylcholine right here. Acetylcholine. Parietal cell, pumping of acid. Acetylcholine also tells the endocrine cells of the stomach to make gastrin, and gastrin is one of those things that tells the parietal cell to do the acid thing, to exocytose the pump and pump acid. So ACH, gastrin. ACH, gastrin tells mast cells, mast, M-A-S-T, T as in Thomas. Mast cells make histamine, and histamine is another positive stimulus for acid secretion. So we have 3 stimuli for acid secretion. Here is the negative feedback. Acid in the stomach tells endocrine cells in the stomach and in the duodenum to make somatostatin, and somatostatin is the inhibitory signal. We're going to see that more when we get to the small intestine. In addition to the nervous system sort of triggering this with acetylcholine, 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 we have some intrinsic gastric phase things, like stretch of the stomach. Stretching of the stomach with a meal triggers gastrin secretion. Putting a meal in your stomach, it stretches the stomach and changes the pH of the stomach. So if your meal has any protein in it, it puts amino acids in the stomach. The carbohydrates can act as a buffer. Changing the pH of the stomach and putting amino acids into your stomach also triggers gastrin secretion. So we have the brain from the cephalic phase working on the stomach, and we have the stomach itself working on the stomach during the gastric phase. All of this is to make gastric acid. Yeah?
Does eating a lot of acidic food have an effect on the negative feedback?
Your stomach produces enough acid. If you drank, I suppose if you drank a liter of lemon juice, something could happen. But it would take something like that to affect the pH. If you ate a whole bunch of Tums, which is an acid neutralizer, it's a bicarbonate acid neutralizer. If you overdose on Tums, that could affect the stomach. But you'd have to eat a lot of it. Here is one of those slides that Bob loves and you hate, but you know how to interpret these slides. So let's go through this. This is a parietal cell. Here is the apical membrane. Here's the basolateral membrane. Here are the receptors for these hormones. So what do we see here? We see that ACH, acetylcholine, from the enteric nervous system and the parasympathetic nervous system, acting at a receptor, activates the GQ story. And now you're off and running, GQ, phospholipase C, IP3, calcium being released from ER, diacylglycerol. Oh yeah, PKC, protein kinase C, is one of the signals that tells the vesicles to exocytose. That's how acetylcholine works. That's how gastrin works. Gastrin is a different receptor, but it activates the same pathway. The GQ pathway, all the steps that you are now sick and tired of, leading to PKC. We're not talking about CAM kinase here, we're talking about PKC here in this story, triggering the exocytosis of the pump. And now the pump just pumps. Histamine works through a GS story, a classic GS story. pKa does the same thing as PKC. Phosphorylate's a different place, but it does the same thing, increasing the exocytosis and apical number of pump proteins. Somatostatin, as you probably could predict at this point, activates GI. GI inhibits the things that GS stimulates, and so somatostatin downregulates the upregulation steps that we talked about earlier. You might predict that the activation steps are more powerful than the inhibition steps because we have GQ, which is not inhibited by GI, and that's probably true. But GI still tends to dampen the production of acid. Let's not worry about prostaglandins. Let's not worry about calcium channels. But everything else on this slide is basic knowledge that you already know. I will not ask you the name of the M3 receptor. I will not ask you the name of the gastrin receptor. I will not ask you the name of the histamine receptor. But you should know that there is a histamine receptor coupled to GS, an acetylcholine receptor coupled to GQ, and a gastrin receptor coupled to GQ. And then you should be able to plug in your GQ story or your GS story. You can't offload those stories yet. So I'm going to skip the next slide because we're going to pick that up after. Oh, no, I'm going to talk about this now. So this is on the last slide. I mean the last page of your handout. Then we're going to go back and do some clickers. We're going to take a break and do some clickers. But this is from your textbook. Here is how we control hydrochloric acid. We've talked about the parasympathetic nervous system. We've talked about, it's more than just the sight, smell, and taste of food. I can make it happen just by talking about food. That's the cephalic phase, but if I brought in here a smell of lasagna, I guarantee that you would be salivating even more. That's the cephalic phase. During the gastric phase, stretch of the stomach, neutralization of some of that acid by the meal itself, and the products of protein all combined through gastrin and the other hormones to increase gastric acid secretion. So you could say, well, what if you eat a meal that is pure carbohydrate and has no peptides? You know, you eat a baked potato with nothing on it. Well, you're still going to do distension, and you're still going to buffer the acid. So 2 of the 3 things will trigger the response of the digestion. You don't have to have protein to get this going. We're going to talk soon about the intestinal phase. But the intestinal phase, when the bolus of food, the bolus of chyme reaches the intestine, we are going to inhibit acid secretion as a way of feeding back. Once the chyme is reaching the small intestine, the body has ways of saying, oh, that meal is now getting into my intestine. That means the stomach can start winding down. And we have a mechanism for that. We'll talk about that in the next section of this class. This is a slide from your book. It's too much. Let's talk about what I want you to know. So we're going to talk, I do want you to know about gastrin. We are going to talk about CCK. We are going to talk about secretin. So that's coming up. I want you to know about those 3. We're going to forget about GIP. They are all peptide hormones. I do want you to know that the site that the gastrin is produced in the stomach. I don't care that you know that it's in the antrum. It's in the stomach. The site of production of CCK is in the small intestine where we are going. And the site of production of secretin is in the small intestine. I do want you to know that. I want you to know the stimulus in the stomach, amino acids in the stomach, peptides in the stomach, and the parasympathetic nervous system are the stimuli for gastrin secretion. That gastrin stimulates gastric acid secretion by the parietal cells. I do want you to know that the stimuli for secreting CCK are the products of protein digestion and the products of fat digestion in the small intestine. And I do want you to know that the stimulus for secretin is acid in the small intestine. The stomach is stimulated by gastrin, and it is inhibited by, we don't have to do both of those. The stomach's secretion of acid is inhibited by CCK, and it's inhibited by secretin. So when those are the negative feedbacks of acid production by the stomach. We're going to get to these other ones when we get to those tissues. So those are the things from this section of the course that I want you to know. Questions about that? So now, we're going to get to a couple of clickers and a break. Before we get to the break, if we weren't so rushed, I would tell an inspirational story about ulcers. I'm going to cut to the chase. Ulcers are wounds or Ulcers are ulcerations, that doesn't help. Ulcers are breakups of the lining of the stomach or the stomach or the duodenum. So injuries in that layer. They can be dangerous because they can be bleeding, and there's a lot of blood flow, and they can cause a lot of problems. There are drugs that are used to inhibit the production of acid as a treatment for ulcers. So histamine blockers, acid pump blockers are used to inhibit acid production to try to calm things down in the setting of a gastric ulcer. Here's the inspirational part. There were two scientists back in the 70s who were starting to discover that it wasn't all acid. There was something bacterial associated with the production with ulcers. And they discovered a bacterium that lives in the stomach called Helicobacter pylori that they said was the causative agent for ulcers. Acid was a part of it, but Helicobacter pylori was causative. And they submitted a paper to the Gastroenterological Society of Australia. This is not one of those societies that you would call high visibility. This is just a regional society of digestive scientists, and it was rejected. It was rejected from a minor society because everybody knew that gastric ulcers were due to acid excess, and this idea that it was due to a bacterium is like, you're crazy. We're not even going to accept your paper. And 18 years later, or is it 28 years? 83, 93, 2003. 22 years later, they won the Nobel Prize for that crazy idea. So the inspirational story is believe your data. Believe, if data are telling you something, persevere. Anyway, so let's go now to there's a clicker, which I'm going to go pass through quickly so that you don't see the answer. Oh, shit. And I want you to do the Davenport diagram for the gastric phase of digestion. We're producing a bunch of gastric acid. Show me, we talked about what your blood pH does. Show me a Davenport diagram that describes that process. Okay, let's take a 10-minute break and work on these few clickers and this one Davenport diagram.